Hi, everybody. Welcome to Forever Green Fridays. Glad to have you all here. Glad to have George and his team presenting. At the beginning, I'm going to start with acknowledging our lovely funders. We've got the Environment and Natural Resources Trust Fund, Department of Agriculture, Clean Water, Land and Legacy Amendment, the McKnight Foundation, and Builders Initiative, among other lovely, generous folks that make Forever Green possible. I also want to start reminding folks that we are on video that's going to be uploaded to YouTube. If you are online, just don't have your camera on. And if you are in person and you don't want your tiny little face to be on a YouTube video, let me know and we'll lure you out. Quick, Mitch is not here today, so I'm going to do the announcements. The only announcement I have is that the Forever Green current round of proposals, we have gotten almost all of our peer review comments in today. I have started to send out the packet of comments to all the folks that I have them for. Everyone else should get theirs. If you haven't gotten it yet, you will get it hopefully by COB today. If it takes a long time, I'll give you extra time. And the PIs will have 2 weeks to respond, which should be 2 Fridays from now. Any immediate questions or concerns on that? You've probably gotten you PIs have probably gotten a lot of emails from me on this already. Which I'm sorry. Colin, did you have an announcement?
Yeah, quick announcement that Forever Green being an innovation platform, right? We have these different functions, right? R&D, commercialization, partnership. The R&D team is in this room right now, and just down the hall, the commercialization team is convening our first all-day gathering of our new small and medium-sized enterprise program, SME program. And I have with me Eddie Benson with the Grain Shed, and he is going to say just a couple words about what we're up to, but small and medium-sized enterprise program is working on, we've enrolled 3 small companies. Teddy came all the way from Spokane, Washington, and is working on bringing Kernza into their business. And so just wanted to keep these things linked up, as the R&D happens, we're down the hall talking about market development. Just want to say a couple words.
Sure. Thank you all so much first for giving us this opportunity to come out and be part of this. I'm from the Grain Shed in Spokane, Washington. We are a bakery and brewery. They're focused on utilizing locally sourced and grown grains, hops, things like that. As many products as we can source locally as possible is what we aim for. And we've had interest in Kernza since before we were about 7.5 years old. And we've had interest in Kernza, but really haven't seen it as something that's accessible up until now, where we're getting this opportunity to start doing some R&D and things like that with it, and hopefully aim to incorporate it into permanent fixtures within our business. So just such an awesome product itself that we're really excited to incorporate into what we do on a daily basis. Thanks.
Are you in communication with the Bread Lab at all?
We do. We have not like a continuous thing, but we do. We are connected through WSU and the Bread Lab and things like that.
Yeah, it's like regional. Yeah, people that have to prep them to that.
Yeah, you see them. We see them at we have grains conferences and things like that around Washington State. Cascadia Grains was what it used to be called. Have worked with them and chatted with them many times.
Yeah, so also in the first SME cohort is 40th Century Grain and the Perennial Promise Growers Cooperative. Can't stay for lab because we have an all-day agenda down there. Just want to keep these things linked up. So thanks.
Great.
Thank you all so much. Really appreciate it.
Yeah, sure.
All right. So thank you all for attending the session today for food science. Just to reiterate that the food science team is myself, Pam, and now Fernanda. We have Levi also joining the group. Today we have 3 presentations. Take, do you want to start sharing? The first presentation is on sake and done by Takehiro Murai. He is currently in Japan. So he's joining from Japan. It's almost like 2:30 here. So, and then after Takehiro, we'll have 2 other presentations, Hazrat and Cecia. We'll be talking about ready-to-eat products and also some meat analog product that we are trying to figure out. So, Take it away. You can do a full screen.
Hi everyone. Thank you, George, for the introduction. My name is Takehiro. I recently finished defending my PhD maybe a month ago or so. I did my thesis on sake production using intermediate wheatgrass. So The broad idea here was can intermediate wheatgrass be used as an adjunct ingredient in sake brewing? And from that broad idea, I broke it down into three parts. The first project was about solid state fermentation using Aspergillus oryzae strains. Aspergillus oryzae are fungus that are used traditionally in sake brewing in the initial parts or initial process of sake. The second project was about brewing the sake with intermediate wheatgrass and then looking at the chemicals, organic acids, aromatic compounds. And then perhaps most importantly, the third project was about the sensory profile and how people perceived those products. And so before I dive into details, I want to introduce what sake is for those of you who might not be familiar. Sake is defined by the National Tax Agency in Japan. It's a alcoholic beverage that's brewed from rice, koji, and water. The koji, this word you'll hear often throughout this presentation, it's rice fermented with that fungus. And then people often ask me, is sake a wine or a beer? To answer that, it's different from both. If you think about beer, you have to first germinate and malt the barley to create those fermentable sugars, and then produce alcohol from it. In the grapes, the fermentable sugars are already there. These two we refer to as sequential fermentation, but in sake, the production of fermentable sugars and the production of alcohol happens at the same time. And so we refer to it as parallel fermentation. This is a kind of a chart of a general, very brief explanation how sake is produced. The first step is to polish the rice. We want to remove the outer layer of the rice. This is because it contains fats and proteins. I'll touch a little more on why we want to remove the fat in sake brewing later. After you polish the rice, you steam the rice, and you take part of that rice and make koji. You ferment it with the mold. Then you take that and unfermented rice, put them together and make a starter mash. And then to that starter mash, you'll usually add a couple additions of more ingredients, more water, creating the main mash. Now, after you ferment the main mash for a couple weeks, then you go through the pressing and pasteurization steps. And so the first project, the solid state fermentation, focuses on the koji making process of sake. These are actual pictures of what koji looks like. It has a kind of a fuzzy appearance to it. And so I tested 3 different strains of koji or Aspergillus oryzae that are one used for sake production, one for soy sauce production, one for miso production. And then, as you can see in the picture, it is known that strains that have high protease activity or production tend to show a greener color, whereas Aspergillus oryzae strains with higher amylase activity or production tend to have more of a pale yellow to white color. And so for preparing the samples, I basically milled the flour and then added water to it, autoclaved it for 20 minutes, and then to that kind of cooked patty, I sprinkled spores of the mold and mixed it, fermented it at 30 degrees Celsius, humidity 65% under draft for 7 days. Typically in the industry, it's only done for 2 days. That's because that's when the amount of fermentable sugars reach the peak. And it's more important for sake brewing purposes. But just for this study, I wanted to see what happens, not just for sugars, but other chemical characteristics for beyond 2 days. That's why I chose 7 days. And then these are the analysis that I conducted for this project: starch, protein, sugar, amino acid, total phenolic, kojic acid, antioxidant capacity, and total flavonoid content. So starch content, And the raw sample or unaltered sample started from about 43 grams per 100 grams on a dry basis. And then as we fermented it towards day 7, starch content reached between 4 to 9 grams per 100 grams. Now, this is not surprising, expected because of all the amylase that are being produced throughout the fermentation. Now, we look at the sugar profile, which is a little more interesting. In the raw sample, very minimal, about 2 grams per 100 grams. But if you ferment it for 2 to 3 days, it reaches total sugar about 27 to 30 grams of sugar. And what's interesting to me from this is if we take a look at the green portion of the graph, which represents verbiscose, it's found in very minimal amounts in the raw samples, but once you heat treat it and also ferment it, it significantly increases. Verbiscose is an oligosaccharide that's usually found in legumes, beans, not commonly found in grains, and so this is why I'm surprised to find it. Verbiscose, they're sold in forms of drugs, pills, that people can take to help kind of the gut health, but oftentimes, sometimes people aren't able to digest it well, and so if we think about whether we can eat fermented Kernza directly, we have to consider about the verbiscose content. And so if we were to treat it as a kind of like a raw ingredient that people ingest, then I would recommend fermenting it beyond the traditional 2 to 3 days, closer to 5, 6, 7 days to minimize the amount of verbiscose. Now looking at protein content, in the raw samples, 18 grams per 100 grams, pretty standard, within the range of value that's been reported for different varieties in the past. And then as you ferment it, it gets closer to 30 grams per 100 grams. This is mainly two reasons. One, just the amount of the amount of microorganisms are growing, and as they consume the carbohydrate from the system, they release it as carbon dioxide, and so there's concentration effect of nitrogen. Then here, amino acid profile. I think a lot going on here. What we can focus on is the lysine. Lysine is a essential amino acid, and is often limiting in cereal grains. And in by fermenting it with Aspergillus oryzae, it alters the profile, the amount of each amino acid, and by day 7, these are the proportions or percentage of lysine present in the entire amino acid group. And so we look at this and we can say that, okay, if we want to have higher amount of lysine in fermented Kernza, then we should be using the methyl strain, in this case, what we found out from this set of strains that we tested. Then we look at total phenolic content. In the raw sample, it started from about 12 milligrams per gram, and then you can see that, is my cursor moving on the screen?
Yeah.
So you can see that if you heat it, it dips a little bit, but then as you ferment it, then it increases back. This again could be because Koji, Aspergillus oryzae, is producing hydraulic amylase enzymes. Phenolic compounds are often bound to carbohydrates and proteins, and so as the enzymes are breaking it, there are releasing phenolic compounds that are then it's going to be a little more easily detectable by the method that we use. Then kojic acid. Kojic acid is a compound that only Aspergillus oryzae strains produce. And in the industry, it's used for skincare products and dental products. It has a property of preventing melanin formation, and so oftentimes they use different substrates, often glucose plates. But I thought it was interesting to see that we can use intermediate wheatgrass as a substrate to create kojic acid as well. Now, I looked at total flavonoid content and antioxidant capacity, which followed similar trends as the total phenolic content that I just showed. And so I'm going to skip those two for time purposes for this presentation. So kind of a brief summary of key findings from the first part of the project. There was presence of verbascose and fermented intermediate wheatgrass, which is interesting because it's commonly found in legumes. And then if you think about the highest lysine production, the miso strain had the highest amount. And then if you looked at the kojic acid production, the sake strain had the highest amount. And then this study has been published online. So for more further details, you can go read this paper. And so that was koji making. Now we're going to move on to the liquid fermentation, the sake brewing part, which I find a little more interesting, more passionate about. Initially, when I started this project, I was hoping to use the koji, the fermented, solid state fermented intermediate wheatgrass as part of the ingredients in sake brewing. But I made it and I smelt it. Immediately, I knew that it was a little too funky for sake brewing. And so for this project, the liquid safe fermentation, I went the route of using traditional rice, fermented rice, and mainly rice, adding a small percentage of intermediate wheatgrass, not the main the ingredients. Intermediate wheatgrass is going to be just a small percentage in this project. So second project. Now I'm going to go in detail of how the sake was produced. So, you take the rice, you steam it. They say different brewers steam the rice to a different moisture content level. Give or take, the moisture content of the steamed rice should be between 30 to 40%, somewhere in that range. And then you add koji, which I didn't, for this rice koji, I bought it online. I didn't produce my own koji. And so you mix the koji, steamed rice, and water. I cooked it at 55 degrees Celsius for 6 hours. This is a very common practice in the industry. This they do this they choose this temperature and time for uh, the reason is that they want to activate the enzymes, but they also, while they do that, they don't want to increase the chance of introducing harmful yeast or harmful microorganisms. So while 55 degrees Celsius isn't the temperature that pasteurization or sterilization occurs, but this combination of 55 degrees Celsius in 6 hours is good enough to prevent or delay natural yeasts or bad bacteria from growing, whilst letting the enzymes work and melt, kind of decompose the starch into the fermentable sugars. Now you take that mash, the hot mash, I let it cool to below 30 degrees Celsius, and I pitch some yeast and lactic acid to create the shubo. Shubo is the mother mash. The yeast that I chose came specifically from the southern island of Japan. Historically, or very, very traditionally, old times, they didn't add lactic acid. They would wait for the natural proliferation of lactic acid bacteria. But in modern times, the approach is you'll add uh, artificial or purified lactic acid to cut down on that time. You don't want to wait for the lactic acid bacteria to form. It takes about 2 weeks. And then I fermented the starter mash for 10 days at 15 degrees Celsius. And then after those 10 days, you add more rice, more koji, more water. Traditionally, on the first day of addition, it's called hatsuzoe. Hatsu meaning first, and zoe means to add. The second day we refer to traditionally as day for odori. We don't add anything. Odori means to dance. I think kind of weird, interesting that they call it a day for dancing when they don't do work. On the third day, they add the second edition called Nakazoe, and then on the fourth day they do the Tomezoe, the last edition. And then after you add all the ingredients, once the main mash has all of the ingredients that you need, I let it ferment for 15 degrees Celsius for 15 degrees Celsius for another 15 days. And then on the last day, I cold crush the mash overnight at 4 degrees Celsius. And then after that I pasteurize it. On the next day I pasteurize it 65 degrees Celsius for 30 minutes. Usually sake is pasteurized twice. Once as the fermentation is done, as they're moving the product from the fermentation tank to the storage tank, they will use a heat exchange pump and pasteurize it then. And after they are ready to bottle it, once they bottle it, they will do the second pasteurization of the bottle. But for this project, I only pasteurized once, which was as I was done with the fermentation, I pasteurized, and I didn't pasteurize what was in the bottle. And this is what it looked like. This is one of the samples. So I had 5 variations, one being control, just rice, and then I used whole intermediate wheatgrass and refined intermediate wheatgrass at 7.5% and 15% inclusion. When I say I included 7.5%, it means that I replaced 7.5% of the rice dry weight. And these are the analyses that I conducted. I looked at the starch, protein, glucose, amino acid, pH, organic acids, alcohol content, aromatic compounds. Oh, sorry, correction, aroma compounds. I looked at the starch and protein content of the raw ingredients that I use. The rice obviously had much higher amount of starch. Refining the intermediate wheatgrass lowered the protein and increased the starch. There's nothing wrong with having high protein in sake ingredients. It just produces a sake with more savoriness or umami. And it comes down to preference. In the sake market, sakes that are a little more fruity, floral, that taste more like wine tend to perform better. And so that is why people often try to use rice with high starch, low protein content. But like I like very savory, rich style sake. So I want to emphasize that there's nothing inherently wrong with using high protein ingredients in sake. But we look at we look at the glucose content. If you could look into the section covered in the box. So we found out that the whole intermediate wheatgrass samples had the lowest amount of sugar towards the later days of the fermentation of the main mash. And one of the reasons here is, or two main reasons. One, the whole samples have less starch available, and so therefore less reduced fermentable sugars available. But also, I mentioned that sake goes through parallel fermentation, not a sequential fermentation. And if we think, if we assume that the whole intermediate wheatgrass has a much higher phenolic compound content due to the bran, then what we can think about next is that the phenolic compounds are known to inhibit amylase activity. And so what we may be seeing is that the conversion of fermentable sugars into alcohol is happening at a faster rate than the conversion of starch into fermentable sugars. This is the amino acid concentrations of sake ingredients. We can see that glutamate, aspartate, arginine were the main components of each ingredient. And then now we look at these are amino acid concentrations of the liquid brewed final sake products. We see that in all samples there's a high amount of aspartate. I looked through different studies that looked at amino acid concentrations of sake, and the profile found in my samples follow very similar patterns to past studies. Um, a lot of past studies report that aspartate and glutamate were the main amino acids found in sake. And the total amount of amino acid usually is reported between 200 to 300 milligrams per 100 ml of sake. And all my samples fell within the range of that. So very, very typical profile found in sake. Here's a graph of the pH. Um, in modern style brewing of sake, it is known that the pH doesn't fluctuate too much because they add lactic acid manually and they prepare the environment that's suitable for the sake yeast fermentation to occur. So no big changes in the pH, which is kind of expected in modern approach of sake brewing. Now we're talking about an alcoholic beverage, so obviously I measured the alcohol content. Um, my control sample, the rice sample, reached 18% alcohol content, which is uh, it's a it's a good sign. It's a sign that fermentation, the whole brewing process was proper, environment was good. Um, the highest that sake, just from brewing, highest alcohol content that sake can reach is about 20 to 21%. That's kind of the biological threshold. Yeast can't tolerate any higher. Um, so I've been talking about this a lot for this project. The very first sake brewery outside of Japan is actually located in Minneapolis. It's called Motoi. It's in Uptown. Uh, and I talked with the brewers there, and they say that their products usually only reaches 18%. And so I would say this is kind of where an how should I say? This 18% is where a brewer can aim for. Reaching higher, closer to 20% range could be very difficult and very technical. Samples that included intermediate wheatgrass were lower in alcohol. What I found interesting was that I expected refined intermediate wheatgrass samples to have more alcohol than the whole samples because they were higher in starch, and so therefore I expected more reducible or more fermentable sugars and more alcohol. That's what I expected. But as you can see in the graph, actually the whole samples had higher alcohol content than the refined samples. And the reason could be is that the nutrients included in the brands, amino acids or minerals especially, contributed to the healthier growth of yeast and therefore produced more alcohol. For the organic acids and aroma compounds, I included two additional samples to the analysis. One is called the Takanobu arrow, I'll refer to as the Taka. And then the second one was the red label. These sakes I chose because I currently work as a bar manager at a restaurant in downtown Minneapolis, and these are the top two selling sakes, and they're readily available at not most, large liquor stores usually have these two sakes easily accessible, and so I chose these samples to analyze. Um, looking at the organic acid, what is interesting is one thing that immediately you can notice that my samples had higher amount of acids overall than the commercial samples. And then specifically, if you look at the blue bars, my samples had a much higher citric acid content than both the commercial samples. And this could be for two reasons. The type of koji I used. It is known that certain strains, certain types of Aspergillus oryzes, asparagus strains produce citric acid, um, which, uh, and then also the specific fermentation environments. It is known that the aerobic or anaerobic uh, amount of oxygen in the fermentation process, entire fermentation process, can affect what kind of acids are produced in sake. Not aware of what kind of lactic acid or what kind of fermentation that the two commercial samples specifically used, and so it's hard to say what exactly differentiates my samples from their commercial products, but we can attribute it to the type of koji used and the brewing process. And then aromatic compounds. These are the conditions used for the gas chromatography. And so I took the results, made a PCA score plot. We can see how each samples are grouped or what's closer to each other. We can see that the samples I made are grouped closely together. Not surprising. Same rice, same brewing process, same brewer. If we look at the taka, it sets apart itself in one direction, and then if we look at the other commercial sample, the red label, it sets itself apart in the other direction. And if we look at the loading plot, which shows specific chemical compounds, aroma compounds, we can see why these samples are grouped together, why these samples are separate. So if you look at the noble arrow, Taka noble arrow, it is characterized by ethyl hexanoate. Ethyl hexanoate, another name for it, ethyl caproate, is a compound that smells like apples, cantaloupe, fruity aroma. And so Takanobu Arrow is characterized as a fruity sake. And then if we look at the red label, a lot of these compounds, especially acetaldehyde, that are found in red label, these are responsible for more of the grassy, herbal, kind of more earthy flavors. And so the red label is characterized by grassy and earthy aroma compounds. And then for my samples, they were characterized by, or specifically the intermediate wheatgrass samples were characterized by ethyl lactate and hexanol. And then hexanol, we can reason that it is because of the fat compounds found in intermediate wheatgrass. I'm going to show some bar graphs. Specifically, I am going to focus on these compounds. I chose these compounds because last year I took the sake sommelier tasting exam, and these are the compounds that you are asked specifically as a sake sommelier to be able to identify. And so going over, again, acetaldehyde has a green, grassy flavor. Ethyl acetate, a little more kind of a sweet, brandy-like aroma. Isomacetate smells like bananas. isoamyl alcohol, similar smell as acetate, but it's more of a dull aroma compared to isoamyl acetate. Ethyl hexanoid again, pineapple, banana, apples. Phenylethyl alcohol is an interesting one I particularly like. It's responsible for floral, specifically rose-like aromas. And so here, the first set of compounds, I want to focus on the isoamyl acetates. So the samples represented by the circled in or boxed in, these are the rice samples. The four samples on the outside of the box are the intermediate wheatgrass samples. And so the rice samples had isoamyl acetate, and then the intermediate wheatgrass samples did not have isoamyl acetate. And so if we think why did this happen, we look at how esters are produced. This visual shows brief transition of leucine to isoamyl acetate. And if we look at the last esterification process, it requires an enzyme called the alcohol acetyltransferase. And towards the beginning of the presentation, I talked about how fats play a role. So it is known that the presence of linoleic acid, which is commonly found in intermediate wheatgrass, the presence of linoleic acid suppresses the gene of sake yeast to produce the alcohol acetyltransferase. And so this is why sake brewers are this is why sake brewers polish the rice to remove the bran, to remove the linoleic acid, so that the sake yeast can produce this enzyme that is key in producing the fruitier esters. Now we look at the ethyl acetate. There's a variation. The red label depicted with the pink bar graph had the highest amount. Among the intermediate wheatgrass samples, the whole intermediate wheatgrass sample, 15% inclusion, had the highest amount of ethyl acetate. And so, oh yeah, so yes, ethyl acetate, the red label is more defined by the presence of ethyl acetate. And then I want to focus on the ethyl hexanoate here. The Takanobu arrow, as you can see, and as I've explained earlier, has much higher amount of ethyl hexanoate. If you look at the intermediate wheatgrass samples, again, the whole samples had the highest amount of, whole 15% had the highest amount of ethyl hexanoate. So wasn't a really trend to whether was it whole or refined, what kind of inclusion there was. There was a clear trend on how these esters amounts changed. So key findings, I think the most important one is that intermediate wheatgrass sake, if you use Kernza in sake production, it specifically lowers isoamyl acetate compared to rice-only sakes due to the presence of linoleic acid. And then whole intermediate wheatgrass causes lower glucose content. As I explained, the reason could be the presence of phenolic compounds, which inhibit the activity of amylase enzymes. And then we also saw that Kernza sakes are characterized by hexanol and ethyl lactate from the gas chromatography. Now this results from this project is limited to the brewing conditions that I used. So let's say we use a much lower fermentation temperature. There could be a whole another profile revealed or whole another profile of compounds that intermediate wheatgrass sake can produce. And so for the third project, I took the samples that were produced in the second project and did a sensory evaluation of it. There is in the US one, as far as I know, one research done by the University of Arkansas. Um, as we all know, Arkansas produces a lot of rice, and they are trying to make Arkansas the sake capital of the US. I want to point out that this study by the University of Arkansas, I think you can maybe see it on the screen, maybe. Maybe not. This study, they used trained panelists. They trained the panelists using the WSET programs, the program used to train wine sommeliers. And so the panelists had an agreed upon definition of linguistics or lexicon, the words that to describe the aroma and taste. They were all trained to the same standard. And then there were there were 11 of them. My participants that the study I did at the bar that I manage, there were 78 participants and they were all untrained. I think this gives us a more better perception of how well the sake products will be accepted if we were to release it. Age ranged from 21-year-old to 70-year-old. Average age was 37-year-old. Then I looked into how much drink in general each participant had. Most people had 1 to 6 drinks or 7 drinks or more per week. I asked participants if they've had sake in the past. Most of them have had sake at least once in the past. And among those who answered yes, I asked them how often do they drink sake. Not often. So my group of people that I were able to have them participate, they've had sake, but most of them don't have sake on a regular basis. That's kind of the profile. I try to look into the answers I collected and see if there were any demographic differences, like say gender, age group, if they had sake before or not. My study didn't have enough distinguishment among demographics of preferences for sake. Two kinds of questions I asked on this study. One was the hedonic liking on aroma and taste. I used a 9-point scale for starting from a dislike extremely to a like extremely. Now, one thing to know about the hedonic liking scale is that it's purely about liking. It doesn't assess how people perceive about intensity of the flavors. Also, the hedonic liking scale, the way the answers or the categories are distant out. This isn't an adequate representation of how people actually. It doesn't connect with people's actual perception, as in disliking something slightly and disliking something moderately. The distance between them isn't always exactly the same of liking something slightly and liking something moderately. It's different for each person, if that makes sense. Another type of question I asked was the label magnitude scale on aroma and taste intensity. And this is what it looked like. And so I was talking about how the categories for the liking scale, they're all spaced out evenly, and so it's not an accurate representation. For the intensity scale, the answers are distant out specifically to it's a logarithmic scale, and so it accurately collects responses of how people perceive the intensity. Now, this is purely about intensity. It's different from liking. Someone can perceive something very intense and still like it. Someone can perceive something very intense and not like it. This is just about intensity. And so the conditions I had, I served it in 2-ounce plastic cups with lid labeled with 3-digit codes. And then it was served at room temperature. Um, Sake or organic acids changes flavor profile at different temperatures. And so if I were to do this project again, I would want to do it at different temperatures. But for this one, I did it at room temperature. I used the Williams Latin square design for deciding the order of samples served to the different participants. Having an extremely sour sake first can skew the perception of all the other samples that come after it. So each participant had a different order of sake to minimize the bias of that. And then I created a kind of online survey on Qualtrics. I had people scan the QR code, answer questions on their phone. And then here, this is the results of the hedonic liking. Let's take a look at one by one. So this one, first one, the Taka, the commercial sample. As you can see, people in general liked the aroma. Most people liked it very much, liked extremely. And then even the taste, people liked moderately or liked very much. And then the red label, this one, the one that smells, the one that's a little more earthy and grassy from the acetaldehyde, it was a little more polarizing. You can see that there's some people disliked it slightly. In general, people liked the moderate very much. As for the taste, people liked it, but you can see a little higher proportion of disliking than the Taka. Then moving on to my first sample, the control sample, you can see that in general, people liked aroma, but if you look at the taste, it's there were some people who did not like the taste. And so you can kind of see like a two humps on the distribution. This is for the whole intermediate wheatgrass sample, 7.5% inclusion. Again, people like the smell in general, but if you look at the taste, there's kind of a more even spread. More people disliked it for the taste. This one, whole intermediate wheatgrass 15%. Again, people like the smell, but the taste was kind of um polarizing. This one was interesting because the panelists didn't know which 3-digit code represent what sample. I knew because I set the code, and a lot of the participants that I came to my study, they were in the restaurant industry, either a sommelier or bar manager or bartender, people who assess wine and sake often on a daily basis, more wine in general. Um, there were memorably 3 people who came up to me after the study. They asked me specifically about this sample, and they said that this sample smelled really good. They asked me, "Hey, I took the survey, it's already submitted, but can you tell me what this sample is? It smelled really good." And so I think it's not an official report, but something to keep in mind. This one for the refined 7.5% inclusion, again, aroma mostly liked, but taste disliked for some people, by some people. The 15% inclusion over here. So because the liking scale, it's evenly spread out, it's not an accurate representation of uh, if liking something extremely, liking something moderately means different for every person. So it's not an accurate, complete representation, but there's 9 points. So if we just arbitrarily make a point system and take the average, the two commercial samples were like the most, the Taka and the Red Label, and then third came my rice sample. For both aroma and taste, the Kernza sake samples performed worse than the rice samples. If you look at the aroma, people like the aroma of the whole samples more than the refined. If you think about the taste, people like the refined intermediate wheatgrass more than the whole. Um, I should have put the bar graph that I showed in the part of the second project. But if you recall, the refined intermediate wheatgrass samples had higher amount of ethyl acetate, and so that could be contributing to higher liking of the aroma, possibly. Or sorry, no, opposite. Yeah. So we can connect this liking to the different aroma compounds present and deduct what kind of smells people prefer. Looking at the intensity scale, the way to look at this violin graph is the red slash represents the mean. The ends of the violin graph represents the range of the responses recorded. So, for example, for the fruity aroma, no one recorded the taka as lower than 4 or 5-ish. Um, So here we can see that people perceived the Taka red label, the rice sample fruity. From the intermediate wheatgrass samples, people perceived the refined 15% inclusion the most fruity. And so here, bringing back the PCA score plots, the Taka commercial sample characterized by ethyl hexanoates, people can pick up on that fruity aroma. Now looking at floral, similar trend where the Taka and the red label and then the rice samples were in general perceived higher intensity for floralness. And among the intermediate wheatgrass samples, the refined 15% inclusion had the highest perception of floralness. Here, the rice aroma. Briefly going back to what I talked about for this project, my panelists were untrained panelists. And I talked about how the study by the University of Arkansas, they were trained to agree to a certain definition of linguistics, lexicon. Um, this again, something that I noticed as people were taking the survey, I can hear people muttering to themselves, "What is rice smell?" And then I realized, oh, okay, so people aren't familiar recognizing or distinguishing what smells like rice. I am familiar with it because I trained to recognize it. Those grassy acetaldehyde smells. But something that I kept in mind was, okay, the results from this rice aroma intensity rating I have to take with a grain of salt. You'll see this in the correlation and p-value data that I will be providing later, but the rating of rice aroma was kind of all over the place. But still, the red label, as I was been talking, distinguished by the acetaldehyde content, had the highest amount, and then followed by the samples that I made. Now we're looking at the taste, the sweetness. People had the people rated Takanobu Arrow as the sweetest sake. and followed by the red label, and then the rice samples. The intermediate wheatgrass samples had the lowest sweetness. And if we look back at the glucose content from the second project, this is kind of in correlation. And so people can perceive the lower amount of glucose. Looking at the sourness, this again, good connection with our second project. The Taka and the red label, the two commercial samples, had much lower perceived sourness. The samples that I made, the rice and the Kernza samples, had much higher perceived sourness. And then if we look back at the organic acid concentrations, my samples had overall uh, much higher organic acids. And we can kind of see why people perceive that. And then we look at the savoriness. The red label, the commercial sample red label had the highest savoriness. To explain this, sake is usually filtered with activated charcoal. Activated charcoal filtration used in water filters, Brita filters. It collects peptides and proteins. Um, Sake producers would do that to make a more cleaner style sake. Um, and I don't know about the Taka, can't say for sure, but the red label, I know for sure that they don't do the filtration through activated charcoal. My samples also, I didn't do activated charcoal filtration. And so then from here, we can assume that sakes that don't go through activated charcoal filtration has a little more peptides and more proteins, and therefore they have a higher perceived savoriness. And we look at the bitterness. Oh, I think this is funny. So the Takanobu Arrow had the lowest bitterness, but as you can see, someone rated the Taka very high in bitterness. It was just one person that kind of skewed the graphic over here, and that was actually Mitch. I remember Mitch was tasting the Taka, and he mentioned, "Oh, this is so bitter." And it was interesting that different people pick up on different notes differently. Yeah. But beside that special case, the red label in my samples had higher bitterness than the Taka. Intermediate wheatgrass samples, especially the whole samples, had higher bitterness. So I talked about the filtration with activated charcoal. Some peptides are known to have a bitter taste. That's also why sake brewers would use charcoal filtration to remove the bitterness of sake. It makes sense that my samples had higher perceived bitterness. Another reason for the whole samples being especially high in bitterness is the presence of phenolic compounds in the bran. And then, so this is a Pearson correlation. This table shows the correlation between the intensity and the liking. So as we can see, fruitiness and floralness had a positive correlation. So the higher the intensity perceived for the fruitiness and floralness, in general, people liked it more. But if we look at the rice, the correlation between the perceived intensity of the rice and the liking, sometimes it's positive, sometimes it's negative, and so it was kind of all over the place. And I was mentioning people, untrained panelists may not be able to pick up on what exactly is rice-like aroma, and so I think this shows a good, like, it shows a picture of that. And here, we look at the taste. the sweetness, perceived sweetness and the savoriness in general had better correlation with liking. Sweetness had higher correlation. Savory in some samples were perceived negative, but here and there, in general, positive, but also not a, if you look at the p-value, it wasn't significant. If you look at the sweetness, it's positively correlated and it's also significant correlation. On the other hand, sourness and bitterness in general was perceived negatively. And so here, conclusion. In general, rice sake samples were preferred over the Kernza samples. I ran some statistics, ANOVA, but it wasn't a significant difference. And then sweetness and savoriness were positively correlated, but bitterness and sourness were negatively correlated. And then fruity and floral aroma was also positively correlated. So this trend of sweetness and savoriness, fruity and floral aroma being positively correlated was also found in the study that I introduced first by the University of Arkansas. And so I found it interesting that whether you're a trained panelist who tastes a lot of sakes, a lot of wines, the general perception of aroma and taste correlated to a liking. Untrained panelists and trained panelists generally have the same reaction. Right now, I'm talking with a brewery in St. Paul. I will be trying to produce sake there.
Hello everyone, I'm Cecia. I will be talking about giving you a high overview about my work. I will be exploring intermediate with grass protein as a novel ingredient for meat analogues production. But why we are doing this? The wheat gluten market is growing exponentially here, especially in North America and also in Europe. And we see the forecast from 2024 to 2024 is going to grow from 11 billion to 26.8 billion, which is a really good opportunity for intermediate with grass gluten. And the highlight in this forecast is US is taking the 42% of this global market, which is almost the half, right? And how is the role of gluten and meat analogues? Which usually gluten is used in meat analogues as a high source of protein and also improve the texture because we want to have this texture like a meat in our product. This is one example how the meat analog look like, even as a product already in the market, how the meat analogs look like. And some application, not sure. Okay. Some application of wheat gluten in the market are in bakery, confectionery, and also nutritional supplement because this ingredient is already established in the market. But why Kernza? If we compare Kernza with wheat, we can see Kernza has a really high protein content, which is around 20, and wheat has 13%. This is a really good advantage if we want to produce a new ingredient. But even though this, we still have some limitation because we don't have any scientific or industrial knowledge how we can extract, how we can process, and also how we can use intermediate with grass gluten. With this in mind, we set some objectives. The objective is develop an efficient method to extract the gluten from intermediate with grass because we want to have this ingredient and also analyze the properties of intermediate with grass to be able to use this ingredient in food application. And as I tell you before, we have a lot of opportunities in the gluten market, right? We want to see which opportunities we can feed there the intermediate with grass gluten as a sustainable ingredient food. And how we will do this? How will we achieve all these objectives? We want to explore two different extraction methods. We want to explore the wet extraction and also the dry extraction by using air classifier. Now we want to use that wet extraction and focus on the gluten proteins. how we will do this? We can do this in the lab. The first thing we are using is the refined flour from intermediate wheatgrass. We will be mixing with water and create this dough. We will rest the dough for some time, and after we will proceed to wash the dough. What is the purpose to wash the dough? Leaching all this starch component and make concentrate the protein in the dough. And after we will dry this and we will get the gluten for the intermediate wheatgrass. Moving on to the dry extraction, we will use a technology called air classification. Now it's used to concentrate legume, for example, pea, chickpea, and also lentil. We want to use the same technology to be able to concentrate the protein. How this technology works? You have an airflow going on, and also you have the flour going, and because the density, different density, you will have at the end into different fraction. The first fraction will be the protein rich, which one we are gonna to use it because we want to use the protein concentrate to produce meat analogs. Which one are some advantage of this? Don't need a lot of energy or water, and also we are eliminating the drying process, which is good. And also we are not using any chemical as other extraction use, which is a really good benefit because we are not damage all this structure of the proteins. And when we get all these two different ingredients, how we can produce meat analog? We have a technology called extrusion, which is a really powerful technique because we can produce different products like cereal, a snack, and also meat. And the process is higher moisture because we have a lot of water in the system. And this is a clear example. If we use soy protein concentrate and with gluten, we have this feeding that's through there, and after we pass through and kind of cooking the product, and we will cool down all this product, and we will get at the end in this nice fiber like a meat. And we compare the two fibers. This one in the right is the meat, the real meat, and the other one is a product. It's, you see, is pretty similar. We are looking for that. We want a product similar to meat. And some challenges we already have, because we already start working on, is if we overwash this dough, we are destroying all this protein structure, and we don't want that, right? We want to get the gluten network to be able to use it. And also, if we add too much water, we are also like solubilize all this protein, and we don't want that. We have some examples in the picture there. And the problem is because the dough for intermediate wheatgrass is sticky, and we compare to the wheat dough, and we want to find a way how we can fix this. And the next step to fix this, we want to develop an efficient method how we can extract the gluten, how we can really make sure we are extracting it the right way, and how we can optimize the way we can drive the gluten and have a good gluten yield and also quality. And after having all these ingredients, we want to evaluate the functional characteristic of this ingredient and be able to use as an ingredient and compare with gluten to be able to see which one are like the different in between other. And at the end, we want to investigate how the air classification of intermediate wheatgrass gluten, how we can use this technology. This is why George is going to the University of Milano to be able to understand how we can use this technology to be able to use the protein fraction of intermediate wheatgrass. And this is all the thing I have for today. Thank you for your attention.